All right, folks. So I broke the four biological macromolecules up into two mini lectures. So 2-9 and 2-10 are carbohydrates and lipids, and then 2-11 and 2-12 will be your proteins and nucleic acids. The reason I did that, not to make your life harder, hopefully it makes it easier, it makes these mini lectures just a little bit smaller and more manageable. So we're talking about the four biological macromolecules, and we'll start out with carbohydrates. First of all, let me just start out with this, a more basic concept. Because the monomers or the small molecules of our body uh, are put together into polymers or these very large macromolecules, organically or organic chemists would have a very difficult time looking at these very large molecules. So what they really look at are the reactive groups on the molecules, and these are called functional groups. And you don't have that many functional groups to memorize. In fact, I wish your book did a better job with them. So there's far more than the functional groups you'll see here, but I'll have you memorize the functional groups you have in your chapter. And we'll just go down through it. An amino group is an NH2 group. This line, sometimes we read it like this, R, NH2. And you say, well, what's R? Anything. What do you mean? What do you mean anything? Anything. R could be any organic molecule. It could be a carbohydrate, a lipid, a nucleic, it could be anything. But an NH2 group is an amino group, and this is what it looks like right here. So NH2 is amino. Amino acids have NH2 groups on them. All right. A carboxyl group is a COOH, and the way it's put together is C double bond O, OH. So we write it COOH, but it's really a C single bonded to an OH, and that same carbon double covalent bonded to an oxygen. So it's the carboxyl group, COOH. And that's also found on amino acids. And in fact, the amino group is the amino part of amino acid, and the carboxyl group is the acid part of an amino acid. It's called a carboxylic acid. So when you say amino acid, you mean at one end there's an NH2 group, and at the other end there's a COOH group. All right, and then there's, by the way, an R group hanging off of there. So that's an amino acid. It has an amino group and a carboxylic acid group on it. Another functional group is a hydroxyl group, also known as an alcohol. And it's found in a lot more than ethanol. Ethanol is your drinking alcohol. An OH group is called an alcohol group or a hydroxyl group. It's found in lots of things. You can see here it's found in carbohydrates, fatty acids, amino acids, found all over the place. The phosphate group is PO4. And the PO4 has a charge. Now it can have, depending on the pH of the solution, physiological pH, the PO4 group usually has two negative charges. Here's one of them, here's another one. That's at physiological pH. It could be protonated, which means you could put an H there, in which case this would only have one negative charge. But that would be an acidic solution that you typically would only see in lysosomes or something like that. And I imagine under certain circumstances you could probably protonate the other oxygen there, and you'd have a PO4. And this, of course, would be an H2PO4, which is not charged at all. But usually, physiologically, this will have a -2 charge because of the pH of our solution being 7.35 to 7.45. All right, carbohydrates. The first biological macromolecule we're going to talk about. Carbohydrates are polymers. They're put together by thousands of polymers. And that's hundreds to thousands of monomers. Well, what is the monomer? The monomer is a monosaccharide. And probably the most ubiquitous monosaccharide is glucose. But fructose is a monosaccharide, and there's other ones like galactose and lactose. So you can, there's other monosaccharides out there. All right. Actually, lactose is a dimer. Lactose is a disaccharide. So I take that back. Lactose is a disaccharide, not a monosaccharide. So your monosaccharides are things like glucose, fructose, and galactose. Those are your monosaccharides. Put two monosaccharides together, you get a dimer or a disaccharide. And probably the most well-known disaccharide is table sugar, sucrose. But there's other ones. There's lactose, which is glucose and galactose. There's maltose, which is glucose and glucose. So there's other disaccharides out there. They're put together by dehydration synthesis. They're hydrolyzed by hydrolysis. This is an example of, I realize there's not hundreds of thousands of here, but glucose residues here, but there could be. When you put a glucose molecule with another glucose molecule, this one glucose that used to be its own glucose molecule is now part of a bigger macromolecule. It's called a glucose residue. Another glucose residue, another glucose residue. Some of these polysaccharides are highly branched, and some of them are not branched at all. Like cellulose is not branched at all, but starch is branched. Glycogen is branched. So different levels of branching there.
Carbohydrates are made of carbon, hydrogen, and oxygen atoms arranged in various ring structures. The disaccharide sucrose is formed in a dehydration synthesis involving the monosaccharides glucose and fructose. When two monomers combine to form a dimer, a water molecule is produced from the dehydration synthesis reaction. Notice which parts of the two monomers were involved in forming the sucrose. The glucose, OH, that gave up its H is the one on the carbon next to the ring oxygen atom when you move clockwise from the oxygen atom. The OH lost from fructose is on the carbon next to the ring oxygen atom when you move counterclockwise from the oxygen atom.
You guys don't have to memorize that. That's a lot of biochemistry. I'm not going to ever ask you where the H and OH came from dehydration synthesis. I just need you to know that dehydration synthesis is combining two monomers together with the formation of water.
When a complex carbohydrate breaks down to simple sugars via hydrolysis, a water molecule is split between the two polymer subunits.
Okay, that's it. Lipids. All right. Lipids aren't built on the monomer to polymer system. So there is no monomer of a lipid that I add thousands together and get this big lipid polymer. Lipids are a diverse class of macromolecules. So why are they all classified as lipids? Because they're all hydrophobic. That's what lipids have in common. They're hydrophobic. The carbohydrates, proteins, and nucleic acids are hydrophilic. And hydrophobic means water-fearing. So lipids are not soluble in water. That's the thing. Hydrophobic is not soluble in water. Whereas my other three biological macromolecules are soluble in water, but not lipids. So we have to talk about lipids with each individual lipid. This is a fatty acid chain, sometimes called a free fatty acid chain. And it's just a and by the way, the hydrogens are left off. They're assumed. When you look at this fatty acid chain, they don't add all these hydrogens in there. They're actually assumed to be there. Carbon always has 4 bonds, so you can put those hydrogens in there. A saturated fatty acid, this word saturated right here, means that the carbons all have a full complement of hydrogens. But you could have an unsaturated fatty acid, and because there's a carbon-to-carbon double covalent bond, this carbon only has 1 hydrogen instead of 2, and this carbon only has 1 hydrogen instead of 2. See, this carbon right here has 2 hydrogens, 1, 2, and this carbon right here has 2 hydrogens, 1, 2. But when you put a double bond in here, you have to get rid of a carbon, or a hydrogen, I mean, because carbon can only have 4 bonds. So this is an unsaturated lipid. Now, who cares? Well, who cares is this. Saturated lipids are solid at room temperature, and they're typically found in animals like humans. Unsaturated fatty acids are liquid at room temperature, and they're typically found in plants. And unsaturated fatty acids are healthier for you. In other words, plant oils are healthier for you than animal lard. So this would be lard, and this would be oil. All right? They are just fatty acids. Well, they're triacylglycerols. They're triglycerides. There's 3 fatty acids per glycerol. But they have fatty acids, and if they're saturated, they're solid at room temperature, and if they're unsaturated, they're liquid at room temperature. As a rule of thumb, it's healthier to have oils from plants that are liquid at room temperature. There is one exception to that, by the way. This is showing you the solid lard, and this is showing you the liquid oil. Okay, let me show you why. Why is this solid at room temperature? Because the fatty acid chains are saturated, and they pack very nicely. There's no bend in them. Whereas when you have a double covalent bond and you're unsaturated, you get a bend in your fatty acid chain. Now these don't stack well. Because they don't stack well, they don't turn into solids. They stay in the liquid form. Whereas these stack very nicely and they turn into solids at room temperature. Now we do have a problem in biology, and that is this. Lipids compose our cell membranes. And there's water outside the cell and there's water inside the cell. Well, lipids are hydrophobic. How could that possibly be? Well, the answer is this. The answer is we make these phospholipids. And the phospholipids have an end that is polar and charged, by the way. This head right here, or end, by the way, there should be a positive charge on that nitrogen. I know it's not there, but it should be. And there should be a negatively charged on that phosphate. I know it's not there, but it should be. This head is hydrophilic because it's polar and charged. Now this tail is hydrophobic. Okay. Well, how do I have a membrane if I have a tail that's hydrophobic and a head that's hydrophilic? The inside and the outside of my cell has to be hydrophilic. There's water inside my cell and water outside my cell. No problem. If I draw this whole molecule like you see right here, here's what my cell membrane is made of. A phospholipid bilayer, where the hydrophilic heads are outside the cell and the hydrophilic heads of the other layer are inside the cell, and my hydrophobic tails are miscible with each other. This is how I make plasma membranes using phospholipids. This is just another phospholipid. It's showing you the details. Here you go. There's your positive charge and there's your negative charge that I promised you was supposed to be there. The picture in your book didn't show it. I wanted to be clear with you that yes, this head is hydrophilic. Another class of lipids are called eicosanoids. Eicosanoids is this molecule right here. It's things like leukotriene, prostaglandin, thromboxane, things like that. Those are eicosanoids. And then we have steroids. Steroids always have these 4 ring structures right here. The 4 ring structure, by the way, is called the cyclopentanophenoanthine. That's what just the ring structure is called. Yeah, what a word for that. You can't play that in Scrabble. That's too long. Cyclopentanophenoanthine ring. That's just this ring structure. And then different steroid molecules all contain that ring structure, but they have different side chains, like double bonded O versus OH. Like this complex chain right here that's seen in cholesterol. Like having a methyl group here, but no methyl group there. So you can see that all steroids are similar, but the side chains coming off them make them different types of steroid molecules. Like cholesterol is a steroid, and then there's some steroid hormones like testosterone and estrogen and things like that. So those are steroids, and these are lipids as well. Here's a table showing you the different lipids. And that's it for carbohydrates and lipids. Next coming up is proteins and nucleic acids.